The lower respiratory system consists of all the structures within the neck and below the level of the neck. These structures are the larynx, the trachea, the bronchi, and the lungs. The lower respiratory system can be functionally divided into the conducting zone structures and the respiratory zone structures. So the conducting zone structures are all of the structures that transport volumes of air to and from the gas exchange sites. So that includes the larynx, the trachea, and the bronchi. Whereas the respiratory zone consists of the region of the respiratory system where gas exchange is taking place. So these consist of the microscopic respiratory bronchioles, the alveolar ducts, as well as the alveoli, the air sacs within the lungs. So let's first examine the conducting zone structures, as they're really a continuation of the upper respiratory system structures as far as the function of moving volumes of air to and from the respiratory zone is concerned. So first we'll examine the larynx, and the larynx, it's also known as the voice box, is a structure that is situated between the pharynx and the trachea. And the larynx is continuous with the trachea that's underneath it. It functions to provide a patent or constantly open airway that remains open even during the pressure changes that occur during breathing. It also functions to route air and food in their proper directions. So it has a structure associated with it called the epiglottis, which closes the larynx when we are swallowing so that food is directed into the esophagus rather than down into the trachea. The larynx also contains the vocal folds, which is why I refer to it as the voice box, and the vocal folds vibrate to produce speech sounds when air passes over them. So the larynx has 9 pieces of hyaline cartilage. That is, these are roughly ring-shaped pieces of cartilage. Some of them are more bar-shaped pieces of cartilage as well. Here we will examine most, but not all, of those 9 pieces of cartilage. So looking anteriorly at the larynx, we see the large thyroid cartilage. It's the largest cartilage of the larynx. It is shield-shaped and it contains a projection called the laryngeal prominence. And this prominence is generally larger in males than it is in females because testosterone stimulates cartilage growth, and it also stimulates the growth of the laryngeal prominence as well. The cricoid cartilage, which is just inferior to the thyroid cartilage, is a small ring-shaped piece of cartilage. And below that, below the cricoid cartilage, is where the larynx ends and the trachea begins. So looking interiorly from the lateral view, we can see the cuneiform, the corniculate, and the arytenoid cartilages, which help to support the epiglottis as well as the musculature and the ligaments of the larynx. And we also see that the arytenoid cartilage is functioning to anchor the vocal folds. So we see interiorly that there are 2 sets of vocal folds within the larynx, or I should say 2 sets of folds within the larynx. The superior set is referred to as the vestibular folds. These are also called the false vocal folds, and the inferior set are referred to as the vocal folds or the true vocal cords or vocal folds. The true vocal folds vibrate when to produce sound when air moves across them up from the lungs. And in living tissue, they appear white in color because they contain elastic connective tissue and they lack any blood vessels on their surface. The false vocal folds, that are the most superiorly located of the vocal folds, do not actually play any role in the production of sound. They function to close the glottis, that is the elongated space between the vocal folds, when we are swallowing. So we see that the vestibular folds are closed in the image on the left during swallowing, which closes the glottis during swallowing so that food does not enter the trachea. On the right-hand side, we see the vestibular folds in the open position, which allows air to move into and out of the trachea. So speaking of the trachea, we'll examine it next. The trachea, which is also referred to as the windpipe, begins at the base of the larynx, and it is a structure that is composed of linkages of cartilage rings, which make it very tough but also very flexible because they are The cartilages are linked together like rings in somewhat like a chain, which gives the trachea a little bit of flexibility. The trachea is approximately 4 inches long, extending to the an inferior point where it splits into two structures that are called the primary bronchi. The primary bronchi are the airways that will enter the lungs. The trachea internally is composed primarily of C-shaped rings of hyaline cartilage lined with epithelia and connective tissues. So we see that there are 4 layers to the wall of the trachea. We have the mucosa, the submucosa, the hyaline cartilage, and the adventitia. So the mucosa lines the lumen of the trachea. That is, the lumen is where the air would be flowing into and out of the lungs during breathing. Deep to that mucosa layer is a region that's called the submucosa. The hyaline cartilage layer is next, followed by an outermost layer that's called the adventitia. So the mucosa of the trachea is lined with pseudostratified columnar epithelium that have cilia on their surface. This is the same tissue that we see in the larynx. Cilia line the surface of the mucosa, and we also see that there are small populations of goblet cells that are embedded within the mucosa, and those function to produce mucus and secrete the mucus onto the surface of the pseudostratified columnar epithelium. This helps to trap dirt, dust, and any debris that makes its way into the larynx. And then the cilia help to move the trapped dust particles and debris particles away from the trachea. The lamina propria, which is the layer underneath the epithelium, is a layer of connective tissue, which is most likely an areolar connective tissue, since that is the connective tissue that we see underneath most epithelial tissues. And it serves to connect the epithelium to the submucosa. We see that smoking inhibits the function of the cilia on the surface of the epithelium, and it eventually destroys the cilia lining the trachea. A lack of cilia means that for chronic smokers, coughing is going to be the only mechanism that's going to remove mucus from the lungs, as the cilia would normally produce the movement for moving that mucus. But without them there, the mucus is unable to move superiorly out of the respiratory system. So the submucosa contains a connective tissue. As we mentioned, it's most likely an areolar connective tissue. And we also see that there are seromucus glands within it that produce tracheomucus along with the goblet cells that we saw in the mucosal layer. So here in this image, we see some of the ducts of the seromucus glands in the submucosal layer, which are also contributing to the mucus production that lines the lumen of the trachea. Examining the hyaline cartilage of the trachea next, we see that there are 16 to 20 C-shaped rings of hyaline cartilage that help to support the trachea as a whole, as well as the submucosa. The cartilage functions to prevent collapse of the trachea during the pressure changes that the trachea is subjected to during breathing, and they also keep the airway open during these pressure changes, but at the same time also keeping it flexible enough to bend and stretch. The rings, as we see, are incomplete posteriorly, which allows the esophagus to expand forward or anteriorly when swallowing food. And we see that between the two parts of the C shape is a band of smooth muscle. It's called the trachealis muscle. This muscle contracts during coughs to expel mucus, and it can also contract to forcefully expel air from the trachea during hard exhalations or during forced exhalations. Moving our way inferiorly down the length of the trachea, we find that the last cartilage, or the inferiormost cartilage of the trachea, is called the carina, and the carina branches to form the right and left primary bronchus, which are the structures that enter the lungs. So we see that the epithelium of the carina is extremely sensitive, so that anything that touches the inside of the carina, like dust, debris, foreign particles, will trigger coughing if they make contact with it. And again, that's a mechanism to help really expel some of that dust or any debris that happens to make its way down the length of the trachea before that material gets into the lungs. So next we will examine the bronchi, and the bronchi are the conducting zone structures that deliver air to the lobes of each lung. So if we are referring to one of them, we would use the singular form bronchus. So for example, in this diagram, we see the left main or the left primary bronchus. But if we were referring to multiple, then we would use the plural form bronchi. So the term bronchial tree refers to the pattern of branching that is seen in the bronchi. The main or the primary bronchi are the branches that serve each lung. So we have two of those. We have a right main bronchus and a left main bronchus. And from there they fan out to the lobar or the secondary bronchi. And the lobar bronchi are serving each of the lung lobes. We see that the right lung has anatomically 3 lobes, while the left lung anatomically has 2 lobes. So there is a single lobar bronchi for each of the lung lobes. The lobar bronchi then branch into the segmental bronchi, which are also called the tertiary bronchi, since they represent the third branching point of each bronchus. We see that the right main bronchus is wider, shorter, and more vertical than the left main bronchus, whereas the left main bronchus is much more narrow, much longer, and also much more horizontally oriented than the right main bronchus due to the position of the heart. So recall that the heart sits along the midline of the body, just deep to the sternum. However, the apex of the heart and the majority of the left ventricle sit on the lower left side of the thoracic cavity, which means that the left lung is going to be smaller because it has less space to occupy in the thoracic cavity. The segmental bronchi, or the tertiary bronchi, continue to fan out as bronchioles. So that is airways that are between a 0.5 millimeter in diameter to 1 millimeter in diameter are what are referred to as bronchioles. There are many small diameter bronchioles that further branch into terminal bronchioles, which are the smallest diameter bronchioles, as these are less than a 0.5 millimeter in diameter and are typically microscopic in size. And these terminal bronchioles are what lead to the site of gas exchange. We see that there are really anywhere from about, depending on what sources or references you look at, there are over 1,000 terminal bronchioles in each lung. Other sources and other texts give us estimates that there are about 6,500 terminal bronchioles in total for both lungs. So that's about 3,000, about 3,200 or so bronchioles, terminal bronchioles per lung. So anywhere between 1,000 and 3,200 is a pretty good estimate for how many terminal bronchioles we see in the lungs. So there's quite a bit of information within this diagram, so I'd like to take some time to review it. On the left in blue in the blue text are the conducting zone structures, and below that in the black text are the respiratory zone structures, that is the site of gas exchange. The upper right part of the diagram shows the various tissue types, and the bars below each one show the relative abundance of that tissue. So the thicker bars mean that that particular tissue is more abundant, and thinner bars mean that that tissue is less abundant. If a particular tissue is absent from that structure, the bar will be missing from that part of the graph. So first we see that in the conducting zone, that is from the bronchi to the bronchioles, we see that hyaline cartilage becomes less abundant as the airways get smaller, and hyaline cartilage is completely absent from bronchioles, as well as the respiratory zone structures like the respiratory bronchioles and the alveoli. But we do see that elastic fibers are present in the bronchioles and the respiratory bronchioles. We see that the elastic fibers are the main support tissue in the bronchioles, that is the bronchioles and the respiratory bronchioles, as opposed to cartilage. We see that there are clara cells that are in the lobar segmental bronchi as well as the bronchioles. These clara cells secrete protective proteins that help to protect as well as regenerate the epithelium that is located within the bronchioles. We also see that the bronchioles have a large proportion of smooth muscle in their walls, which allows the bronchioles to change diameter and control airflow through them. The epithelial tissue changes as we make our way from the trachea down to the alveoli of the lungs. So in the blue structures, that is in the conducting zone structures, the epithelium is a pseudostratified columnar epithelium with mucus-producing goblet cells. In the respiratory zone, we see that the epithelium becomes a cuboidal epithelium, and then it also becomes, even further down in the alveoli, becomes a simple squamous epithelium without any goblet cells present at all. So there is no mucus production in the bronchioles, the respiratory bronchioles, or in the alveoli.